Here's Dr. Kitlinska.
All right, so I want to start from saying thank you. Thank you for allowing me to present our data today, but most importantly to CCF for supporting our research really from day one. It was many, many years ago when we submitted this grant, when the only thing we knew is that Ewing sarcoma have a lot of NPY, and basically that's what it was. And then it was funded, and then it allowed us to do this work, which I will share with you in a minute. But also I wanted to say thank you for organizing this symposium for the scientists as myself, who just spend the time in the lab. This is very important to see the patients, hear the stories. It's always inspiring and humbling. Thank you. So let me start. So as I mentioned, we work on NPY and Ewing sarcoma. So let me guide you through this. So NPY is a small protein which is normally released from nerves. It has only 36 amino acids. There's a lot of it in the brain. It's released from sympathetic nerves on periphery. And then for this protein to act, it has to act through those receptors, which are on the surface of different cells. There are several of those receptors, and then the one in red, I will focus today the most. It's called Y5 receptor. So NPY affects many normal cells, but then also what we know is that many different tumor types have receptors for NPY. And this NPY acting via those receptors does many things which are very relevant to tumor growth. So it can stimulate cell differentiation, proliferation, migration, and so on. So why did we work on Ewing sarcoma? So to start this, Ewing sarcoma can arise in soft tissue and the bones, and it metastasizes most commonly to lungs, but sometimes also to bones. And when it does go to bone, usually it's worse. The outcome is worse. And then it's driven by this fusion protein called EWS-FLI1, which will upregulate many different genes. And NPY and its receptors actually, this is they are EWS-FLI1 target genes. So there's simply a lot of NPY in every single Ewing sarcoma we see. So this is the tissue from human samples, and everything brown on the upper panel is the NPY, and everything brown on the lower panel is the Y5 receptor. So you can see how much of this protein is in those cells. And those tumors release so much NPY that the patients with Ewing sarcoma very often have upregulated levels of NPY in the brain, in the blood, so basic circulating levels. So when we wanted to see what NPY does, we looked a little bit closer at this data. And what we actually saw is that patients who had bone tumors, the primary tumors, had a little bit more of NPY circulating in the blood than those patients with soft tissue tumors. And then another piece of interesting data came from our in vivo studies in mice. So we noticed that the tumors which had a lot of NPY, such as SKAS, this is our model cell line, it releases a lot of NPY. It had a tendency to metastasize to bone, but not to lungs. And then when we looked at the bones within the primary tumor, we saw a lot of bone degradation. So when you look at this bone in the pink on the left, you can see that it's all in pieces, whereas the TC71 xenografts, which don't release NPY, they really did not degrade the bone that much. And another interesting thing was that we made those cells hypoxic, so we deprived them of oxygen. We actually saw even more NPY being released from those cells. So how is this important? So hypoxia is a known metastatic factor in many different tumor types. So we wanted to see how does it affect metastasis in Ewing sarcoma. So we created a model when we made the tumors hypoxic, we deprived them of the blood flow, of the oxygen. And what we saw is that that increased the bone metastasis specifically. So in the SKAS cells, we had more bone metastasis, but not soft tissue metastasis. And even TC71 xenografts, which normally don't metastasize to bone, they also started to produce bone metastasis. So there was clearly a link between this. And there was another interesting observation we made. So when we isolated the cells from those hypoxic tumors, because we wanted to study them, we saw that they look very strange. They were big, abnormal nuclei, and very different in their sizes. But at this point, we didn't know what to make out of it, and I'll come back to this. So just to summarize what we observed, that there is more NPY in patients with bone tumors as compared to soft tissue tumors, that in animal models, those NPY-rich tumors metastasize to bone and they degrade the bone. And both tumor hypoxia upregulates both NPY and bone metastasis. So that really begs the question, is there a link between NPY, these osteolytic features of Ewing sarcoma, and tumor hypoxia? So we started to work on this, and we came out with three mechanisms by which NPY can promote bone metastasis. And that had to do mainly with NPY stimulating this Y5 receptor I mentioned. So what we have noticed that on the right, you can see the Ewing sarcoma cell, which is migrating. And the green signal is the Y5. So you can see how it accumulates in the leading and trailing edge of those migrating cells. So that was very interesting, and that reminded us about a different protein, which is called RhoA. And this is a very important protein for every cell, because what it does, it regulates cytoskeleton. So every time the cell changes shape or moves or divides, this protein is very important in it. So because they really looked very similar, their distribution, we asked the question whether NPY can actually stimulate this pathway. So what we saw that NPY really interacts with the Y5 receptor on those leading edges. So the picture on the lower panel, you can see that those cells which are escaping from the colonies have this red signal, which indicates interactions between Y5 receptor and active RhoA. So the conclusion from this is that NPY acting the Y5 receptor stimulates RhoA, and then when we inhibited, when we knocked down the Y5 receptor in those cells, we saw less cell migration. So the question is this important? So to answer that question, we created an animal model when we injected the Ewing sarcoma cells, either normal cells, which do have Y5 receptor, or the ones which don't have Y5 receptor. And we let the mice to develop metastasis. And what we saw that when we did it, we actually, from the very beginning of the experiment, we could see that our metastasis dramatically decreased, and that actually pertained to all metastases, not only to the bone, so independently of the location. So that was one thing we discovered. But then let's go back to hypoxia. So what we also found that if we make these tumors hypoxic, we actually very highly upregulate this whole pathway, NPY, Y5, and RhoA. And what it does, it actually interferes with normal cell divisions. So what you see on the left, these are two cells which are trying to divide. There are two daughter cells, but because of this high upregulation of this pathway in abnormal time, those cells cannot divide. So what happens, they actually fuse back together and form these big polyploid cells, which have abnormal big nuclei. They are multinucleated. And these are very similar to those which I showed you before we saw in hypoxic tumors. So the question is, are those the cells responsible for bone metastasis? So indeed, when we isolated those cells and we injected them to animals, we saw that you can see on the right dramatic increase in metastasis, and specifically in bone metastasis here in red. And then we saw multiple bone metastases in those mice, which we don't normally see. They are not that common. And again, to prove that this is really Y5 receptor dependent, we created a little bit different experiment. So we also blocked Y5 receptor, but only at the time when the tumors were becoming bigger and then becoming hypoxic. So what happened when we did this, we actually decreased the number of those abnormal big cells, but we also selectively inhibited bone metastasis, so no other metastases were affected. So this was our link between bond metastasis and Y5 pathway. And another thing we noticed is that NPY also stimulates bond degradation. So I showed you this picture before. So the one on the left is the normal SKES xenografts. You can see how dramatically it can degrade the bond. And again, we knocked down NPY in this experiment, so we removed NPY from those cells, and you can see that the bond came back almost to normal. So you can see the very clear border between tumor and the bond, telling us that NPY is important in this osteolytic processes. And what we also noticed is that on the border of the tumor, you can see those purple cells. These are osteoclasts, the cells which degrade the bone. So when we knock down NPY, we also saw less of those osteoclasts. So in summary, what we see is that in the primary tumor, NPY acting via Y5 receptor stimulates cell migration, and that actually promotes overall spread of those cells to different locations. It also stimulates bond degradation. So there's more bond degradation in the primary tumors and also metastasis. And lastly, when we make the tumors hypoxic, we create the overactivation of this NPY-Y5 pathway leads to the formation of polyploid cells, which preferentially metastasize to bone. So what do we do now? So first, we want to see how we can block the effects of NPY in the primary tumor. And it's not that easy. So as I mentioned, Y5 has many different receptors. We know that Y5 is responsible for it. But there are other receptors which can also activate the Y5, transactivate Y5, even if we block it. So now we are trying the combination of different NPY receptor antagonists, see if we can have a better effect. And this is exactly our CCF-funded project right now. We also looked at the bond degradation, trying to identify the osteolytic factors which are released from Ewing sarcoma cells upon NPY stimulation. So far, MMP9, the protease, it's our lead candidate in this process. And lastly, we want to take a closer look at those big polyploid cells which metastasize to bone, to identify the feature which allow them to metastasize to bone, and then to see if we can block those processes. And then we did some characteristic on those cells already, and then there were some features which we expected, but some which we don't expect. So for example, those cells are they start to gain some neuronal features. So when you look at this picture in the bone metastasis, you can see that those cells form those long protrusions and kind of connect to each other. And then one of the most upregulated proteins in those cells is GAP43 here in green, which is known to be normal neuronal protein, is stimulating formation of those protrusions. But also in glioblastoma, for example, it promotes invasion and allows the cells to communicate and avoid treatment. So we are trying to see what is the function of this protein in Ewing sarcoma. So that's everything from me. Big thank you again for all our funding sources, NCCF, for really supporting us for years. And then these are the people behind the work. And I have to say that I have a lot of problems with this picture because it never shows everybody whom I really want to acknowledge. And we are largely student-based lab, so it changed a lot. So I started to add faces first to those pictures. That didn't help. So I decided that we need more pictures then. Thank you.
Okay, so Dr. Kitlinska was very kind in staying on time and even finishing a little bit early, which means there's lots of time for good questions. So if people have a question, raise your hand. Here's one. You should maybe in that.
Thank you. Really nice. I've always just really liked this line of investigation. It's so well done, so congratulations on that.
Thank you.
I have two questions. One is, and I feel like you might have presented this before, so I apologize. What is the source of the aberrant activation of NPY? If EWS fly drives NPY expression, And then in the setting of hypoxia, it goes up even more. Is that like a cooperative activity with HIF, or is it, some other yet to be described mechanism?
So basically, HIF upregulates both HIF-1 alpha and HIF-2 alpha. Further upregulates NPY and Y5 receptor. It's a complicated system, so it completely shifts the activity of NPY from it being activating Y1 receptor, which I didn't mention, but shifts to this NPY-Y5 receptor loop, which is why it's important. And it's further regulated by peptidases, which we try to inhibit, but so it's a complicated system. That's unfortunate.
And then my second question is, what is the source of the hypoxia? You mentioned when the tumors got large, then that's when you assume that there's this induction of hypoxia, but there's also this whole dysfunctional vasculature of Ewing sarcoma. What do you think it is?
So, so this is what we see in xenografts very clearly. So when they start to, grow bigger, the vasculature does not keep up with that. So basically, when we actually did the study when we looked at the tumors at different sizes, and we used hypoxia marker to assess the level of hypoxia. So when you get to over like 100 cubic millimeters in the xenograft, you start to see a lot of hypoxia. This is when we do our hypoxic models, we actually start very early, so to prevent this endogenous hypoxia to take over.
So thank you for the very nice elucidation of this autocrine loop that gets induced by hypoxia. I wondered, you did a lot of loss of function studies, very nice. What about gain of function? Is the autocrine loop sufficient? Or are there other factors?
That is a great question. We did, and it's not. So basically, what we did, we took TC71 cells, we overexpress NPY, and we did not see increase, which basically tells us it's a important but not sufficient. So it's crucial for bone metastasis, but it's not sufficient to trigger it. Which is actually the TC71 story is very interesting, because these are the tumors, or the cells isolated from bone metastasis, I believe, before. And they have abnormal karyotype to start with. But what we notice is that this phenotype of being very metastatic, specifically to bone, it's a transient effect. So if we isolate those cells, we inject into the mice at early passages, they will metastasize to bone. When we grow them longer, they will lose those properties. I think the same happens to TC71, and they need another hypoxic insert to become again.
So that was going to be my second question, is the autocrine loop maintained after the cell is no longer hypoxic? And you're saying no.
So it is. So there was another experiment we wanted to do. So we really wanted to compare, to answer the question whether polyploidy is key, or is it just hypoxic exposure. So what we did, we used flow cytometry, and we isolated different fractions. So we isolated hypoxic 2N cells, diploid cells, and hypoxic tetraploid cells, and we created a progeny. Interestingly, when we were growing this 2N cell, hypoxic 2N cells, they immediately converted to 4N cells. So we could not do this. So that tells you that this effect of hypoxia is a lasting effect, and then it will
Thank you.
cause the changes later.
Yes.
Thank you.
Joanna, I would imagine hypoxia will also increase NPY secretion from non-tumor cells.
Non-tumor cells and normal cells. No, we haven't tested it, but it's actually known. So for example, sympathetic nerves which release NPY, one of the triggers for sympathetic nerves to release NPY is hypoxia. So that's another thing we haven't explored, but I think it's very important. And we are actually working a lot on interactions between nerves and cancer cells.
What I want to come to is, most of these tumors are in extremities, and when the orthopedic surgeons do the surgery, in order to control bleeding, they will put a tourniquet up proximally as they can, and then they do the surgery, and that could be amputation or otherwise. It will be interesting if we can team up with a couple of surgeons and get the recorded time of hypoxia, and if that correlates with long-term survival or metastasis.
That is a great that is a great idea. Yeah. Thank you. I think if you have some contacts, we can certainly do this.
We can definitely hook you up with Dr. Henshaw, so absolutely. Yes. Yes. Microphone in the back. You got it there, Wendy? Okay,
Yeah.
good.
So if I understood correctly, NPY is increased in those Ewings that is in the bone.
Right.
And that usually then metastasizes to bone.
I wouldn't say usual. It's still the most common metastasis are to lungs, and then I think clinicians know more about it. But the bone metastases are more rare, but from our study, it looks like they are driven by completely different mechanisms.
Okay. So you said that there was an antagonist for the Y5R. Do you see more bone degradation because the NPY is still increasing, and you said that was what was causing the bone degradation?
Yes. So actually, I didn't go to too many details about our osteolytic project. And James here is with his posters, you can talk to him. So actually this osteolytic effect, so the bone degradation, it also seems to be dependent on Y5 receptor. So when we block it, we actually see less of the bone degradation. So what we think is happening the same loop, so NPY is released from the cells, activates Y5 receptor, and that also causes release of some kind of osteolytic factors, which will, promote bone degradation. And we are still working on finding out what exactly that is.
So if we can find an antagonist to the Wi-Fi, to the NPY as well, we may have a cure.
Hopefully, we are testing this right now. So we are doing the study. We are testing Y5 antagonists alone and with combination with Y2 antagonists because we think it also contributes. And then hopefully next year we can let you know what is the answer.
Other questions? Did I miss? Okay, so I have a question for you. So let's say that you take a Y5R antagonist into the clinic and you're starting your phase 1 trial and you're going to meet with the physicians and they're going to say, okay, Dr. Kitlinska, we're going to try this new Y5 antagonist, Y5R antagonist in the clinic. What side effects should we look for? So what does Y5R normally regulate or do for cells, and what should we look for in terms of potential side effects?
That is a great question and a very important question. So one of the many things NPY does in our bodies, it stimulates food intakes. So basically when we are hungry, NPY makes us hungry, we eat. So so there are many antagonists developed against Y5 receptor for obesity. So, and that means that when we treat mice with Y5 antagonists, we have to be very careful because they stop eating and they lose weight. Now, the reason for this is because, again, all those pharmaceutical companies were designing those Y5 antagonists to treat obesity, and this is driven by the brain activity of NPY. So all those antagonists we have will cross blood-brain barrier, go to the brain, and stop food intake. So this is definitely something we don't want in cancer patients. So the solution for this would be to make this, change this antagonist so it does not cross the blood-brain barrier. But again, we would need to have some collaboration with the drug design people. And it's possible. We don't have capacity. But when we do experiments, that's what we look at.
Good.
Is there a knockout mouse?
Yes, there is a knockout mouse for everything, NPY, all of its receptor alone and in combination. Again, all done for obesity studies, but it's hard to do the experiment on Ewings because we only have xenografts so far. We don't have genetic model.
But what is the toxicity of the knockout?
Oh, the knockout. Actually, that's a very interesting question. Almost none, because there's a lot of compensatory mechanism. And then there's no effect on development and so forth. So unless you stress the mice, then you start to see phenotype, because NPY is a stress protein, so if you don't have NPY, the stress response is different.
So it's possible that a Y5R antagonist or something with compensatory mechanism and things may turn out to be something with a decent safety margin, a decent therapeutic index.
Yeah.
Let's hope so.
Yeah.
Great. Thank you very much for a wonderful
Thank you so
presentation.
much.